Welcome to The Explainer. Today, we're basically becoming genetic detectives. We're going to figure out exactly how scientists hunt for those tiny, tiny clues, the genetic errors that are hidden deep inside a tumor's DNA.
So where does it all begin? Well, it starts with a single tiny error in our 3 billion-letter genetic code. Just one little typo, a mutation. But think about it. In a sea of that much genetic information, how on earth do you find that one critical mistake?
Well, to find that error, scientists have this really precise 5-step game plan. We're going to start with a tale of 2 genomes to get the basic strategy down. Then we'll see how they prepare the evidence and spot the differences. After that, it's all about filtering out the noise to get to that final report. So yeah, let's walk through it together.
All right, let's dive right into our first section. The whole investigation really hangs on this super clever yet fundamental strategy for figuring out what's gone wrong inside a cell.
So the core idea is actually pretty simple, but it's incredibly powerful. What scientists do is they take 2 samples from the same patient. One is a normal healthy sample, maybe from their blood, and the other is a sample from the tumor itself. It's like having the original, clean blueprint and comparing it to the one with all these suspicious scribbles on it. That's how they spot exactly what's different.
And those differences, those unique typos that are only found in the tumor's DNA, that's what we call somatic mutations. Now, these aren't mutations you were born with. No, these are changes that pop up during a person's lifetime. And these are our prime suspects. They're the genetic culprits that are actually driving the cancer.
Okay, so let's move on to our next section. We're going to see how all this raw genetic evidence gets meticulously prepped for analysis. It's just like a real forensic lab. You can't work with messy evidence. It has to be cleaned up, processed, and organized before you can even think about looking for clues.
And this is pretty much the standard workflow for getting that data ready. Before any of the real analysis can happen, the genetic data has to go through this whole multi-step cleanup process. It's all about making sure every single piece of information is accurate and totally reliable.
And here is the first major challenge. The raw data that comes straight off a DNA sequencing machine, it's a mess. It is just full of noise, low-quality bits, and even leftover digital scraps from the sequencing process itself. If you tried to use it as is, you could end up with totally false conclusions, which is why this cleanup is so vital.
So to deal with that mess, they use these specialized tools. Something like FastQC comes in to assess the quality of the data, and then another tool, fastp, jumps in to basically trim away all the junk. It's like the digital version of cleaning mud off a piece of evidence before you stick it under the microscope.
All right, next problem. A sequencing machine doesn't just read the entire genome in one long strip. That would be too easy. Instead, it shatters it into millions of tiny, jumbled-up fragments. So now you've got all the puzzle pieces, but you literally have no picture on the box to guide you.
And this is where a tool called BWA-MEM is just brilliant. Think of it like a super-powered GPS. It takes every single one of those millions of puzzle pieces and maps it to its exact location on a reference map of the human genome. It's what puts our entire puzzle back together.
Okay, so there's one last and really crucial problem. The process of copying and preparing the DNA in the lab can actually introduce its own errors, and these can look exactly like the real mutations we're hunting for. These are false clues that could send the entire investigation down the wrong path.
To solve that, they bring in the heavy hitters, powerful toolkits like Picard and GATK. These things perform a final super rigorous cleanup. They find and flag any artificial duplicates that were made in the lab, and then they recalibrate the quality scores of the entire dataset. This makes sure that we're only looking at real biological signals, not just lab-created noise.
Okay, with our evidence now perfectly cleaned and prepped, we finally get to the most exciting part of this whole investigation, the direct comparison. This is the aha moment where we finally identify our suspects. What I love about this is that it just boils down this entire complex, multi-step process into one single powerful goal. Everything we've done up to this point, it's all been leading to this one moment of comparison. And the main tool for this job is a piece of software called GATK Mutect2. You can just think of it as our lead detective. It systematically scans both of our prepared DNA files, the normal one and the tumor one, and it meticulously flags every single difference it finds. This gives us our initial list of suspects. But just finding a bunch of differences? That's not enough. our initial list might have dozens, maybe even hundreds of potential suspects. So now we have to interrogate each one to separate the real culprits from the innocent bystanders and the false leads.
Yeah, that raw output from Mutect2 gives us a long list of potential mutations. But from experience, scientists know that a lot of these are just false positives. They're lingering noise or rare technical slip-ups that look like the real thing, but they just aren't.
So to build a truly airtight case, they apply a really strict set of evidence-based filtering criteria. They'll check things like the mapping rate, but maybe the most important thing is the coverage. And coverage is simple. It's just how many times each specific piece of DNA was read and recorded. The higher the coverage, the more confident you are in the result.
And this right here is a perfect example of how strict they are. For a potential mutation in a tumor to be taken seriously, scientists want to see it at least 100 times in the data. 100 times. That gives them incredibly high confidence that what they're seeing is a real biological event and not just some random one-off error.
So after all that meticulous work, the cleaning, the comparing, the filtering, what does the final report actually look like? What's the solution to our genetic mystery?
The entire investigation, all of it, culminates in this one single critical file you see highlighted here, the somatic_filtered.vcf. That VCF stands for Variant Call Format, and you can basically think of it as the definitive rap sheet. It's the final case file listing every single high-confidence mutation that makes this tumor unique.
So this VCF file, that is the case report. It's the genetic blueprint of that one specific cancer, and it's what gets handed off to doctors and researchers. It contains the precise identity of the mutations that are telling that tumor how to grow.
And that leaves us with one final and really crucial thought. This file is so much more than just data. It's the key to that tumor's unique identity. And knowing the precise genetic drivers of a patient's cancer, well, that's the first and most critical step towards personalized medicine, towards developing targeted therapies that can attack the cancer right at its source.